today's episode of Myeloma Crowd Radio, a show that connects patients with myeloma researchers. I'm your host, Jenny Ahlstrom. Now, we'd like to thank Oncopeptides for their support of Myeloma Crowd Radio, because without our sponsors, we couldn't provide such a wonderful program for you. Our program today is a very fascinating one. It will be about the very first CAR T-cell approval in multiple myeloma, and we have waited for this, I think, for decades. So we're thrilled that we'll be able to listen about this program. We are going to start this is a topic that we'll cover multiple times. So, for example, we have an upcoming roundtable where we will also be covering this ide-cel approval on April 24th, just so you're aware of that. We'll be discussing 3 drugs in that, the ide-cel, venetoclax, and melflufen in that particular event. And I would like to start by introducing Dr. Nupur Raje, who is going to be joining us today. So welcome, Dr. Raje. We're so pleased to have you with us.
Thank you so much, Jenny, for having me. This is absolutely an honor. Thank you.
Well, we're so appreciative that you are willing to spend your time helping us understand this better, because it's so new. So let me give you an introduction for you, and then we'll jump into our questions that we have. But Dr. Nupur Raje is a physician-scientist with a focus on the development of innovative therapies for multiple myeloma. She's the clinical director of the Center for Multiple Myeloma at the Massachusetts General Cancer Center in Boston. Dr. Raje leads a dedicated clinical team engaged in investigator-initiated multi-center national and international clinical trials. They're all aimed at developing new and promising therapies for this disease. She also leads translational efforts at Mass General, with her laboratory's efforts focused on identifying cellular signaling pathways that contribute to the survival and proliferation of myeloma cells in the bone marrow environment. She has received her medical degree from the BJ Medical College, Pune University in India, trained in internal medicine at Mass General, and completed a fellowship in hematology and medical oncology in the joint Mass General Brigham Women's Dana-Farber program. She's the first incumbent of the Rita M. Kelly Chair in Oncology at Mass General, and is the recipient of numerous awards, including the Multiple Myeloma Senior Research Award, Leukemia Lymphoma Society Clinical Scholar Award, and the Claflin Distinguished Scholar Award. Dr. Raje, we know that you were very instrumental in some of the early BB2121 clinical trials, and so you're just a perfect person for us to talk to today about this program.
Thank you so much for having me, Jenny. This has been effort on multiple fronts, as you know, and we are so grateful to all the patients who actually participated in this very novel strategy for multiple myeloma, and I could not be more excited that we have this now approved by the FDA, and there's more to come down the pike, and this is really going to open up a whole new way of taking care of multiple myeloma, and it's already here, so this is very, very exciting, and thank you for having me on.
Well, we appreciate you so much. And maybe you just want to give us a little more relevance about what you just said. This is the first CAR T therapy. There will be more. But will you kind of give us a sense of the gravity of the CAR T therapy in myeloma and what this first approval means for patients? Because I think, in my opinion, as a patient, it's a really big step.
You absolutely, you couldn't be more right about this, Jenny. This is for now a first-in-class drug product for myeloma. This is sort of the first immune therapy strategy, and it is the most personalized way of giving treatment. So what is CAR T cells? I'm just going to get started from there, Jenny, because
please.
I think that is important for people to understand. This is where we take out your own T cells from your blood circulation, and once those T cells are taken out, we genetically modify them, insert what is known as a chimeric antigen receptor. That's what CAR stands for, a chimeric antigen receptor that is included onto your T cell, and what a CAR is able to then do is recognize your myeloma cells. Now, in the case of myeloma, most of the CAR T cells which are in clinical trials and are in development are against a protein which all of you are now familiar with. This protein is called B-cell maturation antigen, or BCMA. And the reason to use BCMA is a protein which is essentially expressed on all multiple myeloma cells and on all plasma cells. So the importance of doing sort of targeted treatment is you have to find a protein which is present on tumor cells and not present on other normal cells, because you essentially want to target tumor cells. And that is something we have with this BCMA, and therefore it's not just CAR T cells that are using BCMA as a target, but all of you are familiar with a whole host of other approaches as well. We have, for example, belantamab mafodotin, or Blenrep, which has already been approved. This is a drug antibody conjugate. It has been approved in the context of myeloma. And for CAR T cells, Abecma or ide-cel, which is a BCMA CAR T cell approach, is the first of its kind to be approved in the context of myeloma. So again, a very personalized way of taking care of your disease, wherein we are using your own cells. We are training them ex vivo, that is, training them outside of your body, and then giving them back to you so that they can go and hunt out those myeloma cells and get rid of those myeloma cells and control the disease. So a very novel way of taking care of it, and a very specific way of taking care of patients' myeloma.
Mm-hmm. And can you give us an idea of how long CAR T cell therapy has been in development in general, and then how long in myeloma?
Sure. So, CAR T cell research has been going on for years now. It's going on for more than 15 and 20 years, and it's been specifically helpful in the context of blood-related cancers. So you all know of the approval of CAR T cells for leukemia, for example. It was approved first in childhood leukemia, wherein a CD19 CAR T cell was used, and now more recently we've had approvals in lymphoma as well. So for heme malignancies, once you know that you have a reliable target on your tumor cells, CAR T cells were able to be generated. Right now, we are in the phase where we are using second-generation CARs. So early on, very early on, now this is many years back, 15 and 20 years back, we used the first-generation CARs. That is, we were able to insert this CAR into T cells, and we had what we used to refer to as the CD3 zeta chain. And those CARs were able to do their job, but for a very short period of time. So you were able to activate the T cells. They were able to show some killing, but they didn't last in the body for too long. What's happened over the last close to now more than 10 years is we've started using second-generation CARs. And what second-generation CARs include are you have the chimeric antigen receptor, you're able to recognize the tumor compartment of the tumor cell by recognizing a specific antigen. You have the CD3 zeta chain, which activates the T cells. And in addition to that, we have now inserted what we refer to as a co-stimulatory domain. And what this co-stimulatory domain does is not just activates the T cell, but allows for expansion of the T cells so that they stay in your system for a little bit of time and is able to do the job over time. Remember, we're still very early with CAR T cell development. We are still with second-generation CARs. We have the first second-generation CAR approved in myeloma. Now, in the context of myeloma specifically, Jenny, we've been working on this I'm going to talk to mostly the clinical development. We've been working on it now for the last, I want to say, at least 4 years. With ide-cel specifically, we've treated more than 400 patients now, and there are other CAR products which are being tested, such as cilta-cel and a couple of others which are in clinical trial as well. So still early experience in myeloma, but the drug product or ide-cel or Abecma has been approved based on a study of more than 100 patients. It was 128 patients. And I think before we jump into the details of this, what is to me most remarkable is a couple of things which I do think your audience needs to understand and appreciate. One is, myeloma is an immune disease to begin with. And we, before going into this journey, we were never sure about how effective a patient's T cells are going to be and how amenable they're going to be to us inserting a CAR and inserting those constructs that I talked about and seeing whether or not they would in any way be even functional, especially when you've had myeloma for many, many years and have had continuous therapy for many, many years. Are we even going to be able to generate T cells which are going to be functional, which are going to be able to recognize tumor, and which are going to be able to expand in people after we give them to you? So the starting point, to me, what was absolutely remarkable about even our first in-human study, which we did, I think it was about 3 or 4 years back now, which is when we started, is to see that patients who were getting these drug products, so the CAR T cells, we were seeing complete resolution of disease with complete clearing of bone marrows, which had been unheard of when you're starting to treat a relapse after 4, 5, and 6 lines of treatment. So really remarkable. And as a first step, us able to generate a CAR T-cell product which is going to be efficacious and which is giving patients MRD negative disease state so late in their lines of treatment in itself was, I think, a huge step forward.
It's unbelievable, in my opinion. I mean, I don't know if you've ever seen anything like it in your experience.
No, absolutely never. This was, we weren't expecting this when I first started out. I was a little bit skeptical when I first started out, saying, are we going to be able to see any impact of these T cells? And what we saw was absolutely incredible. In terms of, in the old days, we used to, after giving CAR T cells, do a bone marrow at day 14 and then at 1 month. And those folks who've had CAR T cells know this. We end up doing quite a few bone marrows post CAR T cells. This was essentially learning about this process. And I will tell you, at day 14, patients who had packed marrows going into this modality of treatment, we were seeing complete clearing of that bone marrow. I have never, ever seen this. Even with newly diagnosed myeloma, it takes a little bit of time for whatever treatment you're on for that to start working, and we typically don't even do a bone marrow until you've had 4 or 5 cycles of treatment.
Yeah, it's unbelievable. So to have this now FDA approved, to me, is super, super exciting. You shared a little bit about, so how is CAR T? I just want to start out, I mean, we have a lot to talk about, and this won't be the last time we talk about this approval, just because it's so new and we need to know so much about it as patients, even the very practical things. But can you give us, you said, that it had been used in leukemia and lymphoma. So in terms of like efficacy or impact, how is CAR T different in myeloma versus lymphoma or some of the other blood cancers? What have you seen?
So I think in essence, when you look at CAR T cells, the technology is very similar. The targets we use are obviously different. For leukemia and lymphoma, for example, the target protein that they are focused on is CD19, which is essentially expressed on leukemia and lymphoma cells. We clearly are not doing that, and we're using CAR T cells against BCMA. If you look at responses, the responses are pretty similar across all cancer types, and that is to say that we're seeing very high response rates in excess of complete responses of close to 50% in whichever tumor type you see. What we are not seeing in myeloma compared to, say, leukemia and lymphoma is necessarily a plateauing of the curve in terms of duration of response, because we are seeing patients continue to, patients are relapsing, so it's not something like a cure for myeloma just as yet. But I think, as I've mentioned already, Jenny, this was chapter 1 of CAR T cells in myeloma. And as we start improving upon this, I think we have strategies which we are already doing, is how can we make the duration of response better with a cellular therapy product. So we have to start improving upon the duration of response in our patients, and I think that is the biggest distinction. As far as toxicity is concerned, in myeloma, I will tell you that, again, I was pleasantly surprised. In leukemia, you see a lot more toxicity in terms of CRS and neurotoxicity, but it also may be a question of time. We are sort of the last ones to get approved in myeloma, so we've had the experience of our leukemia and lymphoma colleagues on helping us understand some of the pathophysiology of the toxicity, and we are better at managing it. And so at least with the ide-cel product and even cilta-cel, which is on clinical trials, the toxicity does exist, but it's extremely manageable, and we have tools such as drugs like tocilizumab, which we can use in the context of CAR toxicity. I think knowledge and education around these toxicities has made us recognize them earlier. Having the tools to treat them earlier has also helped us alleviate some of these toxicities and made us more comfortable using these CAR products in the context of myeloma.
Absolutely. And you kind of already talked about why BCMA is being used as a target. Do you want to share early results of some of they call them the KarMMa trials, right? So some of the results from those trials?
Sure. So I will talk about what ide-cel got approved based on. So the first in-human study which we did was a couple years back now, and that's been published. And the new follow-up KarMMa study was, so the first study was about 43 patients. The follow-up study was on 128 patients, and that's what the FDA has based their approval on. Now, when we started the study, we used dose levels all the way from 150 million cells to 450 million cells. The target dose for CAR T cells is going to be 450, but essentially when you look at all comers, the overall response rate was close to 80%, with about 35% of these patients achieving a complete response or a stringent complete response. These responses kept increasing once you got to that target dose of 450 million cells. Now, if you look at all comers, the duration of response is close to about a year, which is in keeping with what we've seen with our first-in-human study, which again is reassuring because first-in-human studies always look better than their follow-up studies. With the KarMMa trial, we were able to duplicate the results of our phase 1 trial. And I think the other important thing to remember here, Jenny, is the fact that this was a multi-center international effort. It was not just done in the United States. It was done across different countries, across the Atlantic, and we were still able to reproduce the same data that we saw with our phase 1 first-in-human study, which is really reassuring because it speaks to the fact that this can be generally applied in centers all over the world, and toxicity can be managed by people all over the world. When you start honing in now on people who actually got either a complete response or a stringent complete response, these patients actually did a whole lot better, where their median progression-free survival or duration of response was 20 months. Obviously, again, that's not a home run, but you have to remember that these are patients who had at least more than 4 lines of treatment. So they'd relapsed after 4 lines of treatment, and the majority of these patients had both bortezomib and carfilzomib, lenalidomide and pomalidomide, and the majority of patients had the CD38 monoclonal antibody, either isatuximab or daratumumab. And those of us who treated patients who had all of these lines of treatment and then have progressed after this, to see stringent complete responses and to see duration of responses in patients achieving stringent complete responses of up to 20 months, again, is quite remarkable in the myeloma space.
Well, right. I mean, a lot of the other drugs have gotten approval, like single-use drugs like daratumumab or Kyprolis or something like that, with a 30% response rate. But you're saying there was 80+% response rate. So, yeah, I mean, you have to think about
the duration
combination.
Of response.
Yeah.
Yeah. I'm so sorry to interrupt, but most of the
ahead.
duration responses with some of these is close to 3 and 4 months. So certainly we made big. It is a big advance, and I think the future is going to be on trying to do even better. The other important thing, Jenny, which I did not kind of focus on, is most of the drugs we use in myeloma, and I think all of you are familiar, once we start you on something, we keep you on that drug pretty much forever. You're on some form of maintenance treatment or what have you. With the CAR T cells, we've given the treatment once, and as of right now, with the duration of response, which I'm talking about, of a year or more, and stringent complete response of 20 months, it's one and done. So you've got your CAR T cells, and you've been on no treatment thereafter, which again, for myeloma, is a huge change in the paradigm of how we take care of multiple myeloma.
Yeah, it's a completely new approach. I think every patient would appreciate a one-and-done therapy. But as you think about these, how we use combination therapies and how you're applying those in myeloma, you haven't even started to apply any idea of combination therapy with CAR T. What's the optimal to do before CAR T? And we can talk about this later in the show, but what's optimal to do prior to CAR T, or when do you do CAR T, and what do you follow it up with? So that is all really unexplored territory.
Absolutely. But that's exactly the place where we are doing a lot of investigation right now. And one of the things, if we've gotten with every drug product that you all have seen in myeloma, typically we first test it in kind of late end stage because we want to make sure that it is safe, it is something which patients are able to tolerate. So again, with the CAR T cells, we've done exactly that. And we've shown that there's safety, and we've shown that there's remarkable efficacy. So the next step then is obviously to move this earlier on. I will tell you that we are doing a lot of different trials wherein we are using it in patients who have high-risk disease. For example, we are using it instead of transplant in a trial. We're using it early on after 1 to 3 lines of treatment in a randomized fashion. We are also using it in patients who relapse early post-transplant as a novel strategy to see if we can actually improve upon the current responses. And you spoke about, right now this is a one-and-done strategy, but with that we're getting disease control for over a year, depending on how the patients are responding. One of our thoughts is, if we do this kind of an approach very late, your T cells are probably beat up. So wouldn't it be nice to do it early? So those trials are happening. And the second question is, if you're getting this response with the one and done, is there a room to sort of combine it with some of the other things? So we are doing those studies as well, wherein to improve upon this response rate, do we need to immune modulate some more so that those T cells can hang around longer and can have better control of disease? All of those are in the setting of clinical trials right now.
Mhm. Yeah, well, that process needs to be run, right? Let me ask you about the sites, because this is not an insignificant therapy to provide, even though you said earlier in the show that the side effects are very manageable now and you know how to do it. But having this treatment at the right site is really important for the first few days especially. So do you want to go into, because I know a lot of patients might think, okay, well, I'm going to go out and talk to my local cancer center and see if they can provide this to me. Can you talk about sites, like how sites are being brought on for this treatment and what's required for this site and what kind of expertise they need?
Sure, absolutely. So, as a starting point, I think the way the drug product is approved right now, the CAR T cells is approved, is for patients who've had at least 4 prior lines of treatment, and then the disease has come back. And that's not an insignificant number of patients. There's a lot of patients who are going to meet the criteria. And typically you've seen an image, you've seen a proteasome inhibitor, and you've seen a CD38. But it has to have been 4 lines of treatment, and that's not uncommon for myeloma. As far as sites are concerned, there are a few basic things which are going to be required, although we're going to try and make this as accessible to patients. So some of the toxicities, and I think this would be a good time to talk about what are the side effects of these and what does it entail, what does creating a CAR T-cell product entail. Well, to start off with, it entails us being able to collect these T cells off of you, first of all, to manufacture the T cells. And that is what we refer to as a leukapheresis process. That means we put you on, those of you who've had your stem cells collected, we put you on a machine, take out your stem cells, and it's usually an outpatient procedure. But prior to that, we give you growth factors so that you can actually spill out those cells. CAR T cells is a little bit different. You do not need any growth factor. We only collect the lymphocytes of the T cells from your bloodstream. It does include being on the machine for 3 to 4 hours to collect those T cells. So it has to be at a center which has the ability to do this leukapheresis, to do the collection. The other thing which we are looking into and which requires sort of basics, and this is the, our stem cells, the ASCT, which is the American Society of Cellular Therapy, or the BMT Society, is now called ASCT. We've changed the name from bone marrow transplant or stem cell transplant to cellular therapy now to include these therapies. But we need a few things for that institute to be accredited. One is they need some kind of FACT accreditation where the transplant societies, you have to meet a certain bar. One is you have to be able to collect this leukapheresis product. You have to have, certain know-how in how to manage toxicities. You have to be able to hospitalize patients because as of right now, they are being hospitalized or they will need to be hospitalized for at least the initial part of their treatment, and then you have to have access to sort of critical care medicine because in a minority of patients you may require ICU support, you might require things like neurological support or things like ventilatory support. So those things should be taken into consideration. And you have to have access to drugs such as tocilizumab. This is a monoclonal antibody against interleukin-6, and we found that it is extremely helpful in patients who develop a toxicity called CRS. I'm going to just take a step back here, Jenny, and I'm going to go over first is the stem cell collection, the leukapheresis collection. Once you collect it, it takes typically anywhere between 3 and 4 weeks for that product to get ready and then sent back to the site. And before we give that product back to patients, we actually give you chemotherapy for 3 days. And this chemotherapy is nothing like what you've had during your transplant, very different. This is chemotherapy called Cytoxan and fludarabine. I think a lot of you are familiar with Cytoxan. Fludarabine is generally new to myeloma patients, but it's given over 3 days. And this can be given as an outpatient. You do not have to be hospitalized for this. Then you get 2 days of a break, and we give you back the cells on the after the 5th day. That is considered sort of your day 0 of getting the cells back. Getting the cells back is almost anticlimactic. It literally takes 2 minutes to push it through a syringe. The cells are, it's not a high quantity, so it's a small amount of cells that we inject into you. The big thing which can happen, though, is because these T cells are very active and because they can go find your myeloma cells and get rid of them quite quickly, they do produce 2 specific toxicities. One of them is called cytokine release syndrome. And that happens when your CAR T cells go find your myeloma cells, get rid of your myeloma cells, and by doing so, your myeloma cells break down. And when they break down, they produce a whole lot of proteins. These proteins can cause a cytokine storm, and that's why it's called cytokine release syndrome. So this is very commonly seen, and if it's not appropriately diagnosed and treated, it can become very it actually can be fatal and life-threatening, and therefore you have to pick the center. You asked about the center. Centers with know-how, centers who know how to manage this, and that comes with experience, obviously, and centers who have the supportive care abilities of ICU support if required. I will tell you we've needed ICU support in the minority of patients, but you have to be ready for that if you're going to be putting a patient through this. And then drugs like tocilizumab, because we know that this CRS responds very nicely to this antibody treatment. So although we want to make it available to all patients, it should be done at centers which have access to all of these facilities.
Right. Yeah. No, it's critical because they know what to watch for. And then you're close to the center or at the center. Is there a certain number of days that you have to be hospitalized for this particular treatment?
So I think that will probably change by product. Right now on clinical trials, we're generally admitting patients anywhere between 10 and 14 days. With the standard of care product, I don't think you need to be in the hospital for the full 14 days. Some few patients may need that, but most patients should be fine for a week of hospitalization. Again, depending on which CAR product you use and what the rate of toxicity, because the toxicities are quite predictable. We could do some. We are also looking into, can we do these outpatient? If you do these outpatient, you have to be very careful and have to really have a center of excellence which understands what they're doing and knowing when to anticipate that toxicity. So certain drug products, for example, Cilta-cel, the CRS happens a little bit later. So if that gets approved, I can foresee seeing that we may be able to do the early part as an outpatient. So these are things which need to be worked out. But again, it would require, at least early on when we're using this drug product, to be used in centers who have experience, in centers who have access to some of the supportive care strategies that I talked about.
And how long will it take for this particular approval, do you think will take sites to come kind of online? Because I know that there's for this particular product, there's a website that's called celltherapy360.com there's like a site locator, Because it's not like, okay, if it's available today, go ask your doctor about it and get started next week, type of thing, right, at this point?
That is correct. But I will tell you that, BMS has done actually a really good job. So I will tell you that my site is up and running, and we've been activated. That may just be that we've done all their clinical trials, so they're very familiar, but they've been proactive about getting sites on board. So I'm sure that website will tell you which sites have access. I haven't yet given a BECMA's standard of care, but I have referred my first patient, so I will see how long it takes. And, initially we always go through a little bit, but I think the process has been ironed out already at places like ours, Jenny, only because we've done a lot of them already. So you do have to go to sites which have it up and running.
Yeah, and then you mentioned the 4 lines of treatment. So I just want patients to understand, because I quite didn't understand this either. When something gets FDA approved like this, they make the FDA approval based on the clinical trial kind of inclusion criteria or what they did. So can you go over you talked about patients with 4 lines of treatment. You had to already get a proteasome inhibitor and an IMiD, and like Revlimid or Pomalyst, and a monoclonal antibody. That is the same criteria as patients who can do this therapy as now a standard of care type of therapy, correct?
That is absolutely correct, and it's really important to see how you count treatments. So typically when we are counting treatments, whatever you've had initially, for example, if patients have had RVD or KRD as their initial treatment, had a transplant and had maintenance treatment, that would not be 3 lines of treatment. That's still 1 line of treatment, Jenny. Then when their disease has progressed, there's 2nd line of treatment, 3rd line, 4th line. So as of right now, they've had to have had 4 lines of treatment. And when I looked at the label, what the FDA has not done is most of the patients, as you know, have been exposed to some of the drugs that I've talked about, but the FDA is not necessarily looking at whether you've been exposed to every one of them, as long as it's been 4 lines of treatment.
Oh, I see. Okay. Well, that's good clarification. Yeah, and like you're saying, I had induction therapy, 2 stem cell transplants back to back because they were tandem. A year of consolidation, a year of maintenance. That was like 1 line of therapy. That's a lot of therapy. So
that's exactly right. So it's important how we count treatments, right? Yeah.
Yeah. If this is your first relapse, then you probably don't qualify for this therapy. That's kind of what you're saying. That's what I want just to emphasize for patients. Yes.
Correct.
Okay. So you talked about the approval for this. You kind of hinted at that other trials are underway, and now they're being used earlier in therapy and with other treatment combinations to increase durability and to increase just depth of responses and duration of responses. We talked a little bit, let me ask just some practical questions. In older patients, because you think about stem cell transplant, I think we're kind of translating sometimes this CAR T therapy. Oh, it's kind of like stem cell collection. We've got this leukapheresis process. It's kind of like a cellular therapy because stem cell transplant is like a cellular therapy. But they're quite different. And sometimes in stem cell transplant, it's not that great for the older population because it's hard on your body. But for older patients, what's the impact on older patients for CAR T? Can anyone kind of do it regardless of fitness status or things like that?
Yeah, so that's a really good question, and I think it's really important to highlight the differences between an autologous transplant and cell therapy, what we are talking about with CAR T cells. Remember, when you do an autologous transplant, you've had your induction treatment, and an autologous transplant or a stem cell transplant is almost to consolidate whatever response you've had from induction treatment. So you're actually feeling your best before you go in for your autologous transplant. You're in a remission, and then you get mega high doses of chemotherapy, which make you extremely sick for the 2 weeks or so, and then you come out feeling extremely well, right? So with CAR T cells, that's sort of reverse order. Most of these, not most, all patients who go into CAR T cells are not in a remission. They actually have progressive disease. They have a high disease burden, and that's the reason why they're getting CAR T cells. So they're in no way in a remission as of right now, and they're being treated for their relapse disease. So we do need to sort of bridge people during this time because they have active myeloma, which is progressing, and we need to control it. The second thing to remember is, we're not giving the mega doses of Melphalan, which is 200 mg per meter square, and you have to be hospitalized. And when you get that dose of chemotherapy, it's a myeloablative dose. When I say myeloablative, it wipes out your bone marrow, and unless you get those stem cells back, your bone marrow would take very, very long to recover if you didn't have that. Now, with CAR T cells, we actually don't give you any kind of myeloablative chemotherapy. We give you just 3 days of chemotherapy, which is outpatient chemotherapy, which is generally well tolerated. And the reason to give that chemotherapy is to make your immune system such that it will be able to accept these activated T cells. So it's not to wipe out your marrow, it's just to regulate your immune system so that we get rid of some of the negative immune effects and your T cells can go and do their work. So again, when you're getting CAR T cells and you get the chemotherapy for the CAR T cells, there's nothing like hair loss, which you see with an autologous transplant. Folks won't even know that you've had CAR T cells if you don't share that with people. You get the CAR T cells back, and as I mentioned to you, the toxicities of CAR T cells are very specific. The two we care about most is CRS and neurotoxicity. You don't see that with an autologous transplant. You can get neutropenia, you can require blood and platelet products, but it's nothing to the level of what you have with the autologous transplant. So quite different. Now, in terms of who are the patients, so certainly, we don't go by age ever for either a transplant or for CAR T cells. But there are certain sort of criteria that people have to be able to meet. And specifically for CAR T cells, we do want your kidney function to be adequate. And the reason for that is when we give you fludarabine is cleared by your kidneys. And when I say you don't necessarily have to have an absolutely perfect kidney function, but you have to have a creatinine clearance of above 30 at least. That's largely because we do need to give you fludarabine, and the data we have is CAR T cells work better when you're able to get fludarabine. So that's one sort of distinguishing factor here. The other few things, when we talk about toxicities like CRS, you have to have a reasonable cardiac status so that you're able to tolerate, if I need to give you fluids, you should be able to tolerate fluids. You should not be somebody who's already requiring oxygen, for example, because then if you end up with a toxicity like neurotoxicity or CRS, that can have really negative impacts. So you have to have a certain fitness level so that you're able to handle the toxicity of neurotoxicity and CRS. Age is not certainly, we've done CAR T cells in people into their late 70s and they've tolerated it fine. I think it's been done in 80-year-old also. But again, it's more the other comorbidities that you have to be careful about.
Okay, I did not know that, so that's great information. Very practical. Now, we've heard about this idea because you look at, who's responded, who hasn't responded to CAR T cell therapy, or who's getting more durable responses. And you mentioned this earlier in the show that sometimes the immune system has been beaten up a bit. Is there any way of developing, or is that in process, to develop an immune system panel? Because you can imagine, this is not an insignificant process, and you kind of want to know, like, is it going to work for me? Is it not going to work for me if I'm going to go do it? So what kind of work is being done right now on creating that immune panel to screen patients, I guess, to say, oh, this would be a great therapy for you. You should go do that.
Yeah, so, the short answer to that is no, we don't have the immune panel. But if you look at the responses, Jenny, we are seeing very high response rates, right? 80% of people responding.
Right.
So I would argue and say that the majority of patients are going to respond. I think what we have to focus on then is what can we do to make that CAR T-cell product better. And I will tell you that we're already doing a lot of work in that space. And what we are trying to understand is in people who have had a great response or where you've had a good expansion of your T-cells, we've begun to appreciate what the T-cell phenotype for that subset of patients should be. And even if you don't have it in your body right now, can we do something to make that better? So there are already trials which are ongoing, and we know that if you have more of a memory T-cell phenotype, your CAR product is going to be better. So those studies are ongoing, and we are also trying to understand when would be the right time to collect your T-cells to make these CAR products. We know that if you are newly diagnosed and you look at your T-cells then versus after 4 lines of treatment, there is a difference in your T-cell function. And what we are trying to do as of right now is understand those differences and figure out whether or not we need to collect early. The other thing we are already doing, Jenny, is we are doing CAR T-cells earlier with the same idea that the T-cells are going to be fitter if we do them earlier. They're not going to be beat up by that treatment, and we'll have to wait and see if that by doing so, it's going to improve upon responses in our patients. We think it will, but we don't know that, and that's why we're doing those studies as well.
And then there are other versions or revisions, like isn't BB2127 kind of a next, like it will continue to evolve, right?
Correct. So BB2117 is getting to that. So what we're doing there is to improve the T-cell function. And what we're trying to do there is, as I mentioned earlier, we already have clues that if you have a memory T-cell phenotype, your expansion is going to be better, your response is going to be better. So BB2117 is a CAR T-cell product which is being exposed to BB007, which then enhances this memory T-cell phenotype. So that's one strategy. Second strategy is doing other ligands. We are using not just against BCMA. Can we do BCMA plus something else? Or can we look at the CAR product to improve upon that T-cell functionality? So all of those things are happening as we speak.
Yeah, it's amazing. I mean, the development that's happening is amazing. Are boosters being considered as well? Like, okay, maybe down the road, a year or something like that, or every 6 months, or I don't know. Or maybe you look at a different kind of immunotherapy as maintenance,
Yeah, so that is also absolutely. So what we have now are sort of umbrella trials where we have one CAR product, and then we have different we're going to have different sort of groups of patients where some will get CAR product alone, some will get CAR product with product whatever else, which we think is going to have an impact versus. So we're trying to do these quickly so that we can get them to patients quickly as well. And those are ongoing studies with all of these drug products. We just don't have the data just as yet as to, which one is going to be the most. And my sense, to be honest, is going to be, again, this is going to be more patient-specific, Jenny. I don't think it's going to be one size fits all. So the more information we get on these drug products, I think what we are seeing with ide-cel right now, or Abecma, is our first foray into CAR T-cells, and I think folks need to stay tuned because, honestly, with this technology, sky is the limit. We're already using healthy donors to create CAR T-cells, so we are not even relying on patient derived T cells, and we have allogeneic CAR T cells which we're using. We're using CAR T cells followed by other drug products, other immunotherapies. You've already seen the promise of bispecific T cell engagers with some of the data out there. And the question is, should we be combining CAR with a BiTE, for example, and seeing if we get more durability with responses? So really an exciting space to sort of test all of this. And for patients, I think it could not be a better time. And I'm so excited to try and get all of these treatments to all of our patients.
Oh, absolutely. And the work that you've done for years now is paying off. So I hope you're proud of what you've done, because we're just thrilled you're doing it. It's really amazing. You've worked hard on this, Joe.
And I can't say enough about patients, because I still remember my first CAR patient where I knew so little about this, but patients go into this with complete trust, and I cannot be more grateful for that. So we can do this by ourselves. This is, it is a team, this, Jenny, and we've always said this, and everybody has played a huge role in trying for us to understand this better and to get it to even more people.
Yeah, absolutely. Well, I want to give some time for caller questions. So if you have a question for Dr. Rajat, you can call 347-637-2631 and press 1 on your keypad if you have a question. While we're waiting for people to do that, I did have an emailed in question that talks about cost a little bit. So for people who are on Medicare or Medicaid, and it might pay for part of it, but maybe not all of it. Do you know how that works yet?
So
because being on study is different, right?
Yeah, no, I know. And on clinical trials, we never had to worry about that too much. But the way it's, my understanding is Medicare is going to pick up the cost for these. And the way to think about this really is, if we look at myeloma and we look at the drug combinations that we use, we started with 2 drugs, Jenny, then we went to 3 drugs. Now we're using 4 drugs, whether it's at new diagnosis or relapse. And again, we're using all of these drugs forever. None of the drugs for myeloma have been cheap. And most of our patients, with the nature of the disease, a majority of our patients have Medicare as their coverage. And we've generally been able to provide care to all patients with help. And I do think that that's exactly what's going to happen, given that this is more of a one and done. So although the cost of it seems a lot as the beginning, if you think about it, and if you're using, for example, RVD as a combination and calculate up RVD's cost for a year, it would amount the same as the drug product for CAR T cells. So I think over time this is going to get ironed out. I would tell patients, we all, at all our centers, have a social worker, a financial person who works with us, and we try and make this as easy as possible, and we negotiate with the insurance. And if we think that this is what's right for you, whoever you're treating, oncologist in the cancer center will help iron out those details. There is going to be a little bit of a learning curve, I think, for Medicare, for the company, and for us as institutions as to how best to try and handle this. But end of the day, I don't think it should be, this is our problem. And given that we are seeing so much promise, I do believe it's going to get worked out.
Yeah, great. Okay. Well, we have a question from caller at 354-0760. Go ahead with your question.
Hello. Hi. Well, thank you, Doctor, for a very informative presentation. I appreciate it. So I'm headed for admission on April 26th for my CAR T infusion. So I was just wondering, I don't know if you touched on, the average durability or what the incidence of durability is. And the other question I have is, what is the incidence of loss of BCMA target? Have you seen much of that?
All good questions. Thank you for your questions, and good luck with your hospitalization. I tell most of my patients it's going to be a boring 2 weeks when you're in the hospital. So prepare yourself and bring stuff to do. You may end up with a fever. That's something, in a strange kind of a way we want to see that because we want to see those T cells expand. As far as durability of response is concerned, on an average, it's a year right now. If you achieve a stringent CR or a CR, I've told you on an average we've seen 20 months and beyond. So, our hope is that we see durable and good responses to this. And in general, as I've mentioned already, it's actually well tolerated, so you hopefully will be very bored with wherever you're at.
I'm praying for that.
There you go, 10 days. And I wish you all the best, and I'm sure you're going to have a great response.
Thank you. What about the BCMA target question?
Yeah, sorry, your question on loss of BCMA. So, I know there's been case reports of BCMA loss, but I will tell you that most times when relapse happens, it is BCMA positive. And that's why, the other question which always comes up is we have so many drug products which are going to be in the myeloma space. I say, hey, more products, the better it's going to be. It's going to be competition. And it's not as if BCMA loss is the primary reason for you to lose response. The primary reason for you to lose response is the T cells start just petering off and they are not around. So giving another drug product down the line would be something completely reasonable. BCMA loss happens in the minority of patients. And if it happens, remember there are other CAR T-cell products that we are testing already, specifically against a new target, and this is called the G protein-coupled receptor 5D, so GPCR5D. Already data with that looks very, very good in the minority of patients who have BCMA loss.
Excellent. Well, thank you so much, and namaste to you, Doctor.
Thank you so much for your question. Great questions. Okay, we have another caller at 309-8893. Go ahead with your question.
Hi, thank you so much. Wonderful presentation. Any sites for patients who have not failed 4 lines of therapy, but, the promise of not being on so many drugs at one time? Are there any centers that are doing any clinical trials prior to failing 4 lines?
Yeah, again, a great question. Yes, there's a lot of ongoing research in the earlier space. And I think the best way to do this would be go on to clinicaltrials.gov and look at CAR T-cells for myeloma. There's trials with cilta-cel. There's trials with ide-cel. We're doing it in patients after 1 to 3 lines of treatment. For example, KarMMa-3 allows you to do that. It is a randomized trial, and that randomized trial, so you don't have the choice of are you going to get the CAR T-cell up front or not. Having said that, the only reason we have it open at our site is if for whatever, so it's a 2:1 randomization, is if you have 3 people, 2 of you will get CAR T-cells, 1 does not get CAR T-cells, and we give them standard of care because it's earlier in the lines of treatment. But after you relapse after that, for whatever line of treatment you're on, you will receive CAR T-cells. So you may not get it up front, and it's almost looking at it as if it's delayed CAR T-cells, but that's one way of getting earlier CAR T-cells. There are other trials with cilta-cel also, which are doing very similar studies, and those would be trying to get the CAR T-cells early. In the high-risk space, we have lots of trials wherein we are doing it even up front instead of a transplant, and that's KarMMa-4, I believe. And there's going to be other studies like KarMMa-7, which are going to test some of what Jenny was asking. What are the combinations that we can start thinking about using in the context of CAR T-cells?
Okay, thank you so much.
And if I can make a comment too, like clinicaltrials.gov is really hard to navigate in my personal opinion. We created a tool on healthtree.org that you can enter like your prior lines of therapy and some of your lab values. And then because all the eligibility criteria is based on that information, and then we have a partnership with SparkCure, so you'll be able to see all the clinical trials you are eligible to join of all kinds, not just CAR-T. But then you can narrow it down and say, okay, I just want to look at the CAR-T trials that I'm personally eligible to join or could be soon eligible to join. And I did that for myself just recently. I spent maybe an hour and a half, 2 hours kind of just digging into the depths, but I didn't have to look through, 450 open trials for myeloma on clinicaltrials.gov. I was looking at maybe 10 or 20. So I could kind of really see, what are the trials and what are they combining, when are they doing it, do I qualify? So I would make that suggestion. Well, Dr. Rajai, we are at time. So I just want to thank you so much for participating on this show. This will not be probably the last time that we invite your participation in a lot of the programs, but your level of expertise in running these studies is so incredible that you were the perfect person to join us for this. So thank you again so much.
Thanks so much for having me, and keep doing all the work you're doing, Jenny. And I'll shout out to HealthTree. I agree completely. It narrows down the clinical trials, as clinicaltrials.gov can be overwhelming for patients. So absolutely. And thank you so much for having me on. Appreciate it.
Well, thank you so much. And thank you to our listeners for listening to another episode of Myeloma Crowd Radio. And we invite you to join us next time to see how we can help connect with myeloma researchers and also contribute to a potential myeloma cure.
Hello, it is Ryan, and we could all use an extra bright spot in our day, couldn't we? Just to make up for things like sitting in traffic, doing the dishes, counting your steps, all the mundane stuff. That is why I'm such a big fan of Chumba Casino. Chumba Casino has all your favorite social casino-style games that you can play for free anytime, anywhere with daily bonuses. That should brighten your day a little. Actually, a lot. So sign up now at ChumbaCasino.com. That's ChumbaCasino.com.
No purchase necessary. VGW Group. Void where prohibited by law. See terms and conditions. 18+.